Sorry, just 1 minute. Can you see the screen? Just 1 minute.
Not yet, sir. Shows that not responding.
I hope you can see now.
It's being shared, sir.
Coming ahead, I think.
The screen is blank. See, right? Yes, yes. Thank you very much, sir.
Okay. I hope you can see my screen now. Can you see?
Yes, sir.
Okay, right.
It's visible.
I'm sorry about the delay.
That's totally fine, sir. Thank you very much.
Okay, now, as it was mentioned, I'm going to talk about the distribution and conservation of endemic flowering plants. As Professor Mayuri mentioned, in addition to various ecosystem services and other benefits, forests also contain a large number of plants and animals. Yeah, very rich, very biodiversity wise, forests are very rich. And also a lot of people know that Sri Lanka is considered as a biodiversity hotspot along with the Western Ghats, and our plants and animals have been studied by many people in the past. And a lot of people talk about only the studies done during the colonial period, but even before that, even in the earlier times, during the king's times, Sri Lankan flora has been studied. In fact, there were lists prepared by people long ago. Now, even in the 4th century, King Buddhadasa wrote Saratha Sangraha, and that contained a list of plants, a compendium of plants. And also in the 8th century, King Agabodhi, King Agabodhi VII, who lived from 766 to 777, compiled a list of plants. Systematic study was done, and all these were actually in Sanskrit. But there are 2 books, Yogarnave and Prayogaratnavali, written by Reverend Mayurapada in the 13th century, and the names were the plant names are given in Sinhala. But then in the 16th century, but by the way, I have to mention that most of the European botanical literature came in the 40th century. The herbers, people who compiled the list of plants used in medicine, those people are called herbalists. Herbalists wrote those herbals. So we started this long before, and then even in Sri Lanka there was a lull period until 16th century, and then only the colonial invasions happened, and then during the time of the Dutch, Paul Herman, a physician attached to the Dutch East India Company, came here, and he lived in Sri Lanka, or Ceylon by that time, from 1672 to 77, and he identified so many different plants. Not only plants, he also collected shells and other things also. and he listed 657 plants in 23 classes. He gave even Latin, Tamil, and single names. Unfortunately, he couldn't publish his work. After his death, William Sherard, he was the director of the Leiden Botanic Garden, published it in 1770, and the name of the book is Museum Zilanic. Unfortunately, we don't have a copy of that book. We only have photocopies, but we don't have the original copy of this monumental work by Sherard based on Hermann's plants. And Johannes Berman wrote Thesaurus Silenicus in 1737, compiling a list of plants with some nine dragons also. And then Hermann's specimens, which were taken to Netherlands, were used by Carl von Linné, who is famously known as Linnaeus. After Latinizing his name, he actually used Hermann's specimens and wrote Flora Zelanica. That was in 1747, before he came up with the binomial nomenclature, which was published in Species Plantarum in 1753. So, what the names given in Flora Zelanica are phrase names, not just binomials. And even the names given in Species Plantarum, published in 1753, are mostly from plants collected by Hermann. So, Species Plantarum, the bulk of the plants listed in Species Plantarum, which were given binomials, were actually Ceylon plants collected by Paul Hermann. And the first superintendent of the Botanic Garden, Alexander Moon, also compiled a list of plants, and name of the book is Catalogue of the Indigenous and Exotic Plants in Ceylon, and that was published in 1824, almost 200 years ago, and that also contains single names. That's a landmark book, although it's not considered as a very good compendium of plants at the time. And then came Enumeratio Plantarum Zelenae in 1850. That was actually compiled by George Henrik Theis, the director of Botanic Gardens, who actually was the director from 1849 to 1880. And he published this Enumeratio Plantarum Zelenae, or enumeration of Ceylon plants. And in 1880, Henry Timon, who was the director of the Botanica Terradinia, published the handbook to the Flora of Ceylon. In fact, it was a 5-volume book. Unfortunately, he couldn't see the completion of the book. He died on the way to a field trip, on a field trip in Kalutara. But then the last 2 volumes were compiled after his death by the then director of Royal Botanic Gardens, Sir Joseph Dalton Hooker. And that was considered at the time that Timon's Handbook to the Flora of Ceylon was considered as one of the classic books of classic floras of the colony. That was supposed to be the best flora compiled ever in a British colony at the time. And then, in fact, the systematic botanist attached to the National Librarian wrote a supplement to that Timon's Flora, and that book came as the 6th volume of the Flora of Ceylon, and that was published in 1931. And then, if you look at the numbers, Timon's Flora reported 1,065 genera and 2,855 species, and 853 species were considered endemic. And then Timon's Flora was obsolete, and Professor B.A. Vikrama with Professor Didasanayake of the Radhiniya started to revise the Flora of Ceylon, the Timon's Flora, and that started in 1968 with funds from Smithsonian Institution, and then the second phase was done from 1990 to 2000 with the funds coming from British ODA. So that included angiosperms, gymnosperms, and pteridophytes. In other words, all the vascular plants were included in the 14-volume Flora of Ceylon, a revised handbook to the Flora of Ceylon. And that included 3,771 species and 926 endemic species. You can see that 853 became 926 in the revised flora. And in 2012, and actually most of the work were obsolete, and there are a lot of revisions and things came since the completion of the revised flora. So the national red list, while compiling the national red list, serious attention was paid to the recent changes in nomenclature, and that list to 2012 national red list reported 3,154 angiosperm species, and the number of endemic species was 894. And in 2020, that was even further revised because some of the species listed in 2012 red lists were found to be not accurately, reported. Some were actually not indigenous, and some were also reported in India. Because now in India also there are a lot of active researchers doing various botanical surveys, and some of the plants we thought are endemic to Sri Lanka now are found in India, in Western Ghats and Eastern Ghats and other places also. So we lost some of the endemic plants, and now they are found in India also. Anyway, now in the latest red list, which was just launched into 2020, there are 3,087 species and 80 subspecies with 885 varieties. So altogether, there are about 950 odd species. Sorry, 3,352 species altogether. Species, subspecies, and varieties altogether form 3,352 species, 52 taxa. And out of those indigenous flowering plants, 863 species, 18 subspecies, and 69 varieties are endemic to Sri Lanka. And a lot of people are little confused when you say endemic, so indigenous and native and all that kind of stuff. But what do you mean by endemic? So endemism is the ecological state of a species. That, I mean, if you say a particular species is endemic, that means that particular species is evolved or originated in that particular geographical location. Whether it can be an island or a nation or a geographical area, any defined place. So if a plant is originated in a particular place, that plant is considered endemic to that particular place, right? So similarly, we have these 950 endemic taxa. When you look at the distribution of these endemic species, you can see that they are not randomly distributed. I mean, most of these endemics are found in the central and the southwestern area. It doesn't mean that endemics are not found in the other areas. So there was a study done using those herbarium records uh, of using the 2012 national release, and there were 85,000 herbarium records, or I mean, nearly 85,000 herbarium records. So those locations were mapped on a grid map of Sri Lanka. In other words, Sri Lanka was divided into 2,847 grid cells, measuring 5 by, I mean, each grid was about 5 kilometer by 5 kilometer, and those localities were mapped on those grid cells. And then there's a measure called weighted endemism. I am not going to be into details of how to calculate weighted endemism. It's a very simple procedure, by the way. And then the degree of endemism in each grid cell was calculated using weighted endemism. And we found that in some areas you have high degree of endemism. So the weighted endemism values are given here. The dark blue ones have weighted endemism values ranging from 12.2 to 21.5, and these yellow ones are the lowest, the areas where you have the lowest endemism. So you can see some areas have high endemism, and the bulk of the country in the north and the east and in some southern areas are not high in endemics. But you can just recognize. I mean, you are able to recognize 9 areas of endemism, or 9 areas where we have a high degree of endemism. And these 9 areas are Central Highlands, here in the middle, and then southwestern wet zone, where you find this lowland rainforest, and northern Highlands, Knuckles and other places, eastern Highlands, Namanukul and similar areas, and then you have Ritigal and Dolukantha, and Bilpattu Jaffna and Yala Hambantut area. So a lot of people think that dry zone doesn't have that many endemics, but we find that the Yala Hambantut, Vilpattu, even Ritical is in the dry zone, and Jaffna, although they are in the dry zone, they are also we can find endemics and rare endemics as well. And in fact, my colleagues, Dr. Kanish Tukul and even Dr. Swama Mika Subra, they told me that even some of the endemic animals show a similar kind of distribution. So we have these 9 endemic areas here in which you find high degree of angiosperm endemics or flowering plant endemics. Now, again, when you talk about endemics, most of the time you think that this lowland rainforest like Singaraj and other areas have the highest endemism. It's not the case actually. The central highlands have the highest number of endemics, and as a percentage, you can see 77% of all endemic plants in Sri Lanka are found in the central highlands. And there are 106 species which are restricted to central highlands. Those 106 species are not found anywhere else in the country, the world. And southwestern wet zone, where Singaraja, Scalian, and other places are, there are 527 endemic species, and that is about 59% of all the endemics in this country. So there are 46 endemic species found only in the southwestern wet zone area. And the northern highlands, that's Knuckles area and surrounding areas, 426 species are found, and that's about 48% of the total endemics in the country. And there are 28 species, 28 species found only in Knuckles area. Eastern Highlands, Namanukul area, there are 230 endemic species, and that's about 26% of all endemics, and 4 species are found only in those Eastern Highlands, so Namanukul areas. Ritigal also has 3 species restricted to Ritigal, and those 3 species are found only in Ritigal and nowhere else on earth. So that's about 10% of all the endemics in the country. Yala doesn't have any peculiar species confined only to Yala, but Vilpattu and Jaffna each have 1 species confined to those. Like Vilpattu has 1 species and Jaffna 1 species. Those are found only in Jaffna. Those flowering angiosperm species. Uh, so the percentage wise, they don't. I mean, they are less than 10%. Ritical has 10%, but Dulukanda, Yala, Vilpattu, and Jaffna endemic areas are having 6%, 5%, 3%, and 2% respectively. If you look at the relief map of Sri Lanka and compare that with those endemic areas, we can see that it has something to do with the elevation also. Now, even here, the southwestern wet zone, we see some areas we have higher elevations, and in the middle, the central highlands, the eastern highlands, and northern highlands are also, as you can see, they are raised to high elevation. Elevation is high, and percentage endemism is also high in these areas. Now, in those 9 endemic areas, there were 5 areas. Within those endemic areas, there were 5 areas which has the highest number of endemics. So, high degree of endemism. So, they can be considered as co-endemic areas. Now, as the degree of, if you rank them according to the degree of endemism, Adams Peak comes first, and then comes Singaraja, third Porton Plains, fourth Knuckles, and Candy. So Adams Peak, Singaraja, Porton Plains, Knuckles, and Candy can be considered as co-endemic areas. We have the highest concentration of endemic plants in the country. And if you look at the numbers, Adams Peak has 510 species, and there are 20 species which are found only in the Adams Peak area, nowhere else on earth. And that's about 57% of the total endemics. And Singaraj has 406 species, and 5 species are confined only to Singaraj. You don't find them anywhere else. And the total number of, no total percentage of endemism in the Singaraj area is 45%. That comes second actually. The highest is Adams Peak. And Horton Plains also has a high number of endemics, 302, and there are 25 species which are confined only to Horton Plains. As you can see, Horton Plains has more unique endemics even compared to Adams Peak. Adams Peak has 20 species only in Adams Peak, The Horton Plains has 25 species, which are only found in Horton Plains, but that's a very small area. About 2,000 acres, hectares. And then within that small area, you find this many endemic species, and that's about 34% of the total number of endemics in the country. Knuckles also has 12 endemic areas only found in Knuckles area, 33% of the total endemics, and Kandy. When you say Kandy, I mean, it's not the Kandy city, but there are surrounding areas like Hantana, Calais, and Mandala. There are surrounding areas which are not actually urbanized. Still, we have some vegetation left in those special areas in Kandy. Unfortunately, one of the endemic species is considered extinct, and the last reported locality is Kandy, that's Sobilanthus caudatus. So these are very important when it comes to conservation. So, as we all know, angiosperms, like any other plant, are threatened by many factors. Out of those 863 endemic species, 625 are threatened. When I say threatened, they are listed as either critically endangered, or vulnerable. And that's about 72.5% of the total angiosperms in the country. And if you look at the subspecies and varieties, 13 out of the endemic subspecies and 13 out of the 18 endemic subspecies and 50 out of the 69 endemic varieties are threatened. So if you pool them up, if you add them all up, the endemic taxa, species, subspecies, and varieties, the total is 688. So we have a total number of endemic taxa 688, and that's if you look at the total number of endemics, it's about 950. If you look at as a percentage, 72% four are threatened. Now, I told you that there are 950 endemic species, 950 actually endemic taxa, because 863 endemic species and 18 subspecies and 69 varieties. So if you add them all up, the total comes up to 950. So that's endemic taxa, endemic angiosperm taxa. If you add endemic Pteridophyte taxa, Pteridophytes we have 42 species and 3 endemic varieties, 42 endemic species and 42 endemic varieties, sorry, endemic subspecies of Pteridophytes. So if you add them up with the, if you add that to the endemic angiosperm taxa, 950 plus 45 comes to 995 taxa, vascular taxa, because angiosperms and dendrophytes, they are both vascular plants. And then we don't have a single conifer indigenous to the country. We only have two gymnosperms, two cycas species, and we don't have any endemic cycas species. So only dendrophyte can be considered as vascular plant. I mean, other than angiosperms. So altogether, we have 995 vascular taxa endemic to the country up to now. But then I know there are about 5-6 varieties, species, being described. There are 2 or 3 papers already accepted. Some, and some are yet to be prepared. And I know there are more than 5 new species, new endemic species to be described. And if you add that, those also, the total number of endemic total number of endemic vascular taxa will be little over 1,000. But still, we are not qualified to become a biodiversity hotspot of our own, because to be eligible to be called a biodiversity hotspot, you have to have 1,500 vascular endemic vascular species. But since we don't have 1,000, more than 1,500, we are listed as a biodiversity hotspot along with Western Ghats, because we only have 900 are not vascular endemic taxa. Anyway, the message here is that out of those endemic species or endemic taxa, 72%, 72.5% are threatened. So that's an alarming thing. But when you look at the distribution of those threatened plants, because now that earlier map I showed you, which showed the endemic areas were recognized using weighted endemism. If you do the same thing for the endemic, for the threatened plant, you can see almost the same pattern. But there are no threatened plants in Yala, or Vilpattu, or Jaffna endemic areas. But you have threatened endemic plants in Reticula, Dolukanda, Northern Highlands, Central Highlands, Eastern Highlands, and Southwestern wet zone. So if you overlay this map or the threatened plant distribution map with the endemic plant distribution map, you see something like this. Yeah, uh, there are so many endemic plants and also so many threatened plants. So if you combine threatened plants with the percentage of the high degree of endemism, you can say that these species are very important when it comes to the degree of endemism, when you consider the degree of endemism, and also they are very important as there are a lot of threatened plants. So we can see that six endemic areas: Central Highlands, Southwestern Wet Zone, Northern Highlands, Eastern Highlands, Critical and Dolukanda are, I mean, we can't use the same term hotspot, but something like hotspots because there we have high degree of endemism plus large number of threatened plants. And so those threats may be due to various factors. I mean, obvious thing is the habitat loss, destruction of forests for clearance, quick clearing these forests for various activities may be the primary factor, the driver. But then there are other factors like environmental pollution, and then indirect factors such as unavailability of pollinators or dispersal agents. Now we know now if you take bees or even other insects, the bees are, or if you take the bambara bee, Apis dorsata is considered as the most effective pollinator in our region. So what we do is a lot of people set fire to bambara hives and also chase away bambara. So even do a lot of harm to the insects who carry out pollination. And then if you don't, protect those pollinators, they won't pollinate. I mean, they won't be pollinators. And one other factor which a lot of people don't consider is the impact of the invasive alien species. So many of those invasive alien species have very attractive flowers, very attractive because they are fragrance or their amount of pollen or the nectar and various things are so attractive. The pollinators come to those invasive alien species, leaving behind the native species. So the native species are waiting for the pollinators, but pollinators go and pollinate the invasive species. Yeah, and that's bad in two ways. One is that the invasive species will spread, and their population will get increased. And also at the same time, the native plants will not get pollinated. Then there will be a new generation coming from those native, endemic, and other indigenous plants. So there are those factors are there. So even though there are forests intact, still we can see a large number of species are endangered. So those factors may be, influencing those degree of threats, and also dispersal agents. So there are now if you take some animal, some birds. Now there's a study conducted by Chaminda Vijayasundara of Peradeniya University, and he found that the endemic bird populations have got increased in the recent past in the mountain area. So probably because those endemic species may feed on the increased number of invasive species. Because the invasive species have also got increased, and since they are feeding on those, plants, maybe their numbers are increased. They are feeding and also they are pollinating. Now, if you take Sesper morontiacum, an invasive plant, it's pollinated by hill whiteye and dispersed by yellow-eared bulbul. Both birds are endemic. So there are various things which we can see happening, and those drivers are affecting the conservation of our native endemic plants. And so there are various reasons: the habitat loss, lack of pollinators, dispersal agents, pollution. We are spraying chemicals and things like that. So all these factors may be leading to those losses, and we can see the number of the number of the percentage of threatened plants has got increased. Now, if you look at the number of the percentage of threatened plants, threatened endemic plants, in the 2012 red list, we found that 66% of all endemic plants were threatened. But now in the 2020 list, we found 72%, 72.5% of the endemic plants were threatened. So in the 2012, it was 66.3%. In the 2020 list, is 72.5%. So we can see how these things are changed. So that shows we have to be careful and we have to take action. The policymakers and other people should do some policy level changes, and also we, the normal people, the average citizen, should do our part to conserve our important endemic, because those endemic plants, those 1,000 endemic taxa are found only in this country, very small area, but within our very small country, we have that many 1,000 endemic taxa only found in our country. So that's the message, and thank you very much for inviting me to give this thematic talk, and I wish the symposium will be a very successful one. So those are my references. That endemic area paper we read in 2016, and the second one is referring to the national red list. Thank you very much. Acknowledge Mrs. Dakshani Perra of the Environment Ministry, who did the GIS analysis for the endemic area work, and the staff of the Wildlife Secretariat, director and the staff of the Wildlife Secretariat of the Ministry of Environment, and the members of the expert groups and the red listing team at the National Herbarium. Thank you.
Thank you very much for that those interesting facts, sir. Would anybody like to ask a question from sir? We don't have much time, but maybe one or two.
Yeah, no problem.
All right, so in the absence of questions, I would just like to know a small thing. As you said, there's a lot of, there are some endemics in Vilpattu and Jaffna. Is there any particular reason as to how they manage to stay isolated from India, given the proximity to southern India? So,
no, actually, you mean they haven't gone?
No, yeah, exactly, yes.
So these are mostly herbs, and then they were actually confined to thick jungles, and then maybe now if those are to cross the sea, bird or I mean wind or something, but there are no dispersal agents, and then they are not carried away, carried the long distance through wind. So actually, the speciation rates are not that high. That's one reason why we don't have that many species. And of course, there are a lot of disturbances also, especially in the central, north central area. But I should also emphasize the fact that large areas of the north central province are not properly botanized. And if you seriously botanize some of those areas, if you look at the Google map also, you see many of those areas are thick jungles. And the story may be a little different. I'm not saying that. that many endemic species, but you might find many other endemic species in those areas as well.
Okay. There's one question in the chat box, sir. What is the point endemic species in Vilpattu? I suppose on plants.
Yeah, I can't remember, but I mean, if you can email, my email address was given there, and I can send it to you. Offhand, I can't remember.
Okay, thank you very much, sir, again. So can we then move on? And as he said, the email address is there, I suppose, in the in your proceedings document. Can we now move on to the technical sessions?
Yes, sure, madam.
So is our first presenter SSK Sadani? Are you logged in? SSK Sadani, I presume you're the presenter. No?
Madam, till she joins, shall we move on to the next
one?
Yes, madam.
So there's DEN Mandavala. Are you?
Yes,
okay, okay. So can we proceed with yours? So your presentation, DEN Mandavala, is on the correlation between pupil color dimorphism and gender ratios of Papiliopolytus romulus in Sri Lanka. And the presenter is Eranda Mandavala. The floor is open. We can see your presentation.
Good morning, ladies and gentlemen, and respected panel of judges. My name
Can we request you to speak a little louder?
I'm going to talk about correlation between pupil color dimorphism.
Can you speak a little louder, please?
Madam, it's a recording that I've sent.
Okay, okay. All right, sorry. Okay, then you continue.
Judges, my name is Eranda Mandavala, and I'm going to talk about correlation between pupil color dimorphism and gender ratios of Papiliopolytus romulus, also known as the common mormon, in Sri Lanka. This butterfly is one of the most common and most widely distributed swallowtail butterflies found in Sri Lanka. It is also found in other Asian countries such as India, Singapore, Bangladesh, and so on. Females show polymorphism existing in three forms: Romulus, Cyrus, and Stichius. This map shows the distribution of common mormon in Sri Lanka, and as you can see, it is found in all climatic zones. This table shows the identified larval fruit plants of common mormon in Sri Lanka. There are 14 larval fruit plants, all belonging to the same plant family. This graph shows the seasonal abundance of common mormon in Sri Lanka, and you can see in November it shows the highest seasonal abundance, and the lowest seasonal abundance is seen in July and August. The immature stages of common mormon in Sri Lanka have been described by these authors, and they have also observed the pupil color dimorphism. However, detailed information on duration and measurements of immature stages were not found in literature in Sri Lanka, but found in Singapore, Bangladesh, and India. Therefore, the objectives of this study was to determine correlation between pupil color, gender, and gender ratio of common mormon, then to record the duration and measurements of immature and mature stages of common mormon, and finally to observe factors affecting pupil color plasticity. Coming to the materials and methods, a female common mormon was observed laying eggs on a lime plant in my home garden in June. 16 eggs were found, and once they hatched, they were collected with the leaves and reared in the lab of Horizon campus. They were placed in a glass container of dimensions mentioned here, and on a daily basis, fresh lime leaves were provided and frost was removed. The duration and measurements of each stage were recorded, and photos were taken using a DSLR camera fitted with a macro lens. Towards the end of the final insta, sticks were placed in the glass container to encourage the larvae to climb onto pupate. Once the adult butterflies emerged, their wingspan was measured, their sex was determined, and they were all released back into the wild. The immature, mature stages and larval fruit plants were identified using these various published reference sources. Moving on to the results discussion, these images show the immature stages of the common mormon. The first image here shows the egg. The second image shows the mature egg. These two images shows the newly emerged larvae feeding on the eggshell. This is the first instance, second, third, fourth, and fifth. So in each stage, the duration and measurements were recorded, shown in this table. So all 16 eggs developed to adult butterflies. So the mortality rate in this study was 0%. Since all adult butterflies were released to the wild, their duration were not recorded. These images show the pre-pupa and pupal stages. This is the pre-pupa. Pupa of common mormon is observed in two colors, the brown color which is shown here and the green color that is shown here. This is the brown mature pupa and the green mature pupa. These images show the adult stages. These first two images show the male, open wing and closed wing, and the female, form Romulus, open wing and closed wing. Out of the 16 pupae, we observed 12 were green and 4 were brown, which shows green pupa dominance. Both type of pupa gave rise to both males and females. Therefore, we saw no correlation between pupal color and gender. As far as the gender ratio goes, 12 of the 16 adult butterflies were male, that is 75% male dominance, and all 4 females were form Romulus. This gender dominance has also been observed by these authors in India. They report that a batch of eggs collected from a single plant on a given season showed either male dominance or female dominance up to 80%. But they do not mention whether these eggs are from a single female or what type of larval food plant or which months of the season showed male or female dominance. So there is some incomplete information, but in our study we have observed a male dominance of 75%. Environmental factors influencing pupal color plasticity have been studied in India, and various factors have been identified and are mentioned here. Out of these, the substrate texture was found to be the most dominating factor, followed by background color. In our study, 92% of the green pupae were made on the wall of the glass container, that is a smooth substrate, and all 4 brown pupae were made on sticks, that is a rough substrate. So this shows a strong correlation between pupal color and substrate texture. Coming to future studies, the effect of season on pupal color plasticity can be a subject of future study, because this butterfly is found in varying abundance throughout the seasons, and the type of larval fruit plant on pupal color plasticity also can be studied, because we have observed only in lime fruit plant, but it has 14 identified larval fruit plants in Sri Lanka. And also our study is a captive study. In a non-captive setting, these results can vary. This butterfly is also found in all climatic zones. Therefore, any variation of pupal color plasticity in different climate zones can also be investigated. In conclusion, we can say that we can report from this study there was no relationship between pupal color, gender and gender ratios. And also we can report that in the month of July, eggs from a single female on a lime plant showed male dominance. Further, we can say that a strong relationship between pupal color and substrate texture was observed. These are my references. Thank you.
Thank you very much. The floor is open for any questions now. Eranda, can I ask you one question? So your objective was to look at the effects of environmental color on what exactly was your objective?
The objective was to determine if there's a relationship between the pupil color, because there are two pupil colors.
Yeah, I know, green and brown, right?
Yes.
Yeah.
Find out if there's a relationship between the gender and the pupil color and or the gender ratios. That's the main objective.
Okay. But you say the pupil color is influenced by the environment, right? Surrounding environment.
So from that, the substrate texture was the most dominant factor. So that was also observed in our study, because in the twigs, the sticks, the brown sticks, the brown pupa were made, and the pupa that were made on the wall of the glass container, that is a smooth surface, was green color.
Yeah, that leads me to another question. Now, if it's the texture, now both, if you say the glass will be transparent and smooth. The sticks will be rough and brown.
Yes.
So how can you conclude whether it's the color or the texture that affected it?
Yes.
And the other thing is that, and this is just a food for thought for you. If you're going to conclude, then the availability of both should be the same, no? Because if there was more glass surface than the availability of sticks, of course they will go to the glass surface. How can you explain that? If I say, okay, they stuck on the glass not because of the texture or the color, it was because it was more extensive than the sticks. Do you get my point?
Yes, but it's, we can't control it, isn't it? Because the larvae will choose whether it will not want to pupate on the glass or the stick or any other surface or anything.
Yes, I know that's exactly. I mean, the larvae may choose because of the availability.
Yes.
Isn't it?
Yeah.
So, now, if you had sticks painted in, I don't know, the paint might be some way of putting a natural color. If you had natural green, if you had green sticks, the similar sticks painted in, or the brown ones, and you somehow got it colored green, then at least you evade the textural difference.
Yes, that's true. Yes, correct.
Yeah.
So the background color was the second most dominant factor that was found in literature. So
in the literature, that's true. But from your conclusion, I don't know whether you can conclude. Firstly, it's these might also have some genetic component, and you're only looking at one female's clutches. Plus, if your target was to look at the effect of color or texture, then the experimental design would have to address that, isn't it?
Yes, I understand.
Yeah, so in future, I mean, this is constructive criticism, and I'm supposed to be asking questions, so since the others didn't. Does anybody have any other question? All right, then thank you very much, Eranda, right? Eranga. Can we move on to Thadani, Thadani's presentation? Ms. Thadani, are you here? All right, then we'll go to the next one. SST Samarasinghe, are you here?
Yes, madam, I am here.
Okay. So here we have a presentation on the examination of the effectiveness of environmentally sensitive areas in conservation of biodiversity outside protected areas in Sri Lanka. So it will be presented by SST Samarasinghe. What's your first name? Asugandhi.
Sugandhi.
Right, over to you, Sugandhi.
Thank you, madam. Good day, everyone. Here the research topic is about examination of potentiality of environmentally sensitive areas in conservation of biodiversity outside protected areas in Sri Lanka. Sri Lanka is a biodiversity hotspot among other 36 biodiversity hotspots of the globe and exhibiting a remarkable biological diversity and is considered the richest country in the Asian region in terms of species concentration. The statistics given in the IUCN Red List in 2012 and 6th National Report published recently show the evidence of the endemicity and the wide array of ecosystems ranging from diverse ecosystems in the country. In this scenario, conservation and management of biodiversity is essential for a sustainable development because the population pressure, increase in demand on natural resources, and environmentally insensitive planning and implementation creates the biodiversity depletion. However, the country harbors protected area around 26.5% of the land. However, there is a significant extent of biodiversity outside these protected area networks. It is evident when looking into these maps. The left-hand side one shows the protected area coverage of the country, which is managed by the Department of Wildlife Conservation and Forest Department. And then these maps are few examples where it shows the endemicity of butterflies, amphibians, odonates, and the mammals. So it clearly shows these endemicity or range restricted species are not confined to the protected area network. In this scenario, what are the solutions we have to conserve this important biodiversity? OECM, otherwise other effective area-based conservation measures, is a emerging concept which has been promoted with the IG targets and been validated in 2018 and defined in Convention on Biodiversity and by IUCN. If we look into the IUCN definition, which is more simpler, it is conservation designation for areas that are achieving the effective in situ conservation of biodiversity outside of protected areas. There comes two important words, which is sustain and long term. Again, IUCN and CBD recognizes four broad governance types for the protection of protected areas. This could be governance by the government, private sector, or diverse right holders or stakeholders, or lastly, indigenous people and the community of the area. Having this broad concept with the literature, the objective of this study was to see the potentiality of environmentally sensitive area concept as a OECM, which is an area-based conservation approach used in conserving biodiversity sustainably, and qualitative methodology had been used with the case study approach, where primary data has been used from key informant interviews and Gangewadi ESA co-management plan, whereas secondary data came from records of official sources and was assessed against the three criterion on sustainability, whereas participation, voluntary commitment, and conservation action. Look at this map. Look carefully. The land uses show 75% of this area is forest and agriculture is for 15% and water bodies are 10%. This is a part of Vanathavilluwa Divisional Secretariat Division, and if you look carefully you would see this end is Vilpattu National Park, whereas in the southwest area there is the Veera Kodichole Forest Reserve, and in the northeast there is the Barif Marine Sanctuary, and this area belongs to the Ramsar Vilpattu cluster. So it shows this area which is actually around 2000 hectares is really important. If look further, it revealed in a baseline survey done in 2017, there are around 921 species within this small area, and 10% of them are endangered while 6% of them are endemic. How come? It is because this small area actually harbors very important ecosystems. It ranges from terrestrial ecosystems to brackish water, then coastal ecosystems. So in these sensitive ecosystems, it harbors this important species diversity, and it was obvious that gene diversity also very high, and there were the evidence of wild rice varieties. The findings and recommendations were interesting, and multidimensional stakeholder participation, which was very important, referring the government, community-based organization, private sector and academia, who actively participated for the identifying biodiversity within diverse ecosystems, and then the available threats, mitigation measures and resources. Different needs and interests of different communities, irrespective of their age, gender, and other diversities, are being visible in the planning process for benefit and burden sharing. The voluntary commitment of all stakeholders has been inbuilt through the rules in the co-management plan, including transforming from harmful practices to the sustainable practices with resourcing and monitoring the progress of conservation. The co-management plan indicates that all activities are aimed at the reducing threats and in situ conservation of biodiversity with associated ecosystem functions and services through various approaches. Finally, the study reveals that ESA is an OECM which is having the potentiality to conservation of biodiversity through conscious building for the conservation culture. Thank you all.
Thank you very much, Sugandhi. The paper is open for any questions or clarifications. Please don't leave it to me only. So yes, Sugandhi, can you just tell me what was the basic method you used to see the effectiveness?
Yes, madam. Actually, there we use this sustainability criteria. It is about when it comes to the sustainable development that with the participation and
what do you mean by we can hear you. What do you mean by participation?
It is about actually engagement of these stakeholders, because there were really a lot of stakeholders with us, including from the divisional secretary himself, divisional secretariat, Department of Wildlife, Forest Department, then Central Environment Authority, communities, private sector, all of them, so that when they engage in the decision-making process, in the actually from the very beginning, identifying biodiversity, identifying the threats, then identifying these threat mitigation measures, and developing the management plans, and then in the implementation. So that was one criterion. Then the voluntary commitment of them. So because this actually not came by the mandate itself, because the mandate was in a way limited to Forest Department or Department of Wildlife Conservation. But then here in this case, all the stakeholders were willing. So their voluntary commitment was really important. Then again, conservation actions. When we were looking into the actions in the management plans, we realized actually it was supporting a lot, because they are in a way that we worked, these people have worked in a changing certain practices. So those actions then again supported in minimizing the threats. So that's how these actions, these criterions were used for measuring the effectivity. But it is, it needs a long time, but it is actually took a short while, but this is has to be a continuous study also.
Yeah, thank you very much. A small, any other questions? If not, I have another small question. Yeah, just to find out now, this is all amidst the ESA is surrounded by Vilpattu, then another forest under the Forest Department, then the Ramsar site is there. All of it is there. How sure are you? Say if there is another ESA now, this is all we know. This area is very much, the Kalaway Basin area is very much on every conservationist agenda, so to speak. But if this ESA was demarcated or designated in another isolated area, what would you think? Would it be different, or would it be the same amount of commitment and the same amount of engagement and all that? What do you think?
It could be context specific, but then again, based on the importance of the biodiversity and the ecosystem services, and then how it relates to the people's livelihoods and how it relates to the sustainable development, we would expect that there could be some similar enthusiasms. Actually, that this research showed that we could see certain similar models in other places also, like in Anuradhapura, in Maneva Kand area, then again in Galneva area, so that also there is potentiality to see this in other places also. So here that even there are my other colleagues, other authors who participated here, join in this discussion, and I invite them also to add more points in this regard.
Okay, thank you very much. I think we will move on to the next. Yes. Because of the lack of time. All right, can we? Yes, please. We can't quite hear you. You have a question? Hello. All right. So then there is our next presentation lined up is on the diversity and composition of benthic macroinvertebrates in the Putlam Lagoon, Sri Lanka. And our presenter will be N.D. Hettige. So over to you.
Okay, thank you, madam.
It's best if the others can mute the mic.
Good day, everyone. My title is Diversity and Composition of Benthic Macroinvertebrates in Putlam Lagoon, Sri Lanka. Introduction. Benthic macroinvertebrates are invertebrates organisms visible to the naked eye that live in aquatic water bodies and in or near the bottom of the aquatic sediments. They are considered as successful indicators. Also, benthic macroinvertebrates play an important role in aquatic ecosystems as food sources. Also, the ability to promote mineralizations, mixing of sediments and cycling of organic matter. Puttalam Lagoon is the third largest lagoon and one of the productive lagoons which is located in the northwestern province of Sri Lanka. The lagoon is fed by two rivers, namely the Kalaoya and Niyoya. It is linked to Mundal Lagoon 15 kilometers to the south by a canal. The lagoon's water is brackish to saline. The lagoon has a surface area of 700 hectares and the surface temperature of the water ranges from 28 Celsius to 32 Celsius. Seasonal precipitation levels are driven by monsoon winds and shower bimodal patterns. The long rains fall between mid-May and early October, and the short and weaker rains fall from December to early March. There are numerous aquatic biodiversity studies in this lagoon. However, benthic macroinvertebrates in the lagoons of Sri Lanka is not comprehensively discussed. The main objective of this study is to identify the diversity and composition of benthic macroinvertebrates in the Puttalam Lagoon. Methodology. The study site is Puttalam Lagoon in Sri Lanka. The various anthropogenic activities may cause indirect impact on water quality. Sampling location. Sampling was carried out in April 2017. This is the sampling location map. 16 sampling locations were selected. Analysis of samples. Sediment samples were collected using GAT sampler. Collected sediment samples were preserved using 70% of ethanol. Sediment samples were wet sealed. After separation, benthic macroinvertebrates sorted. They were examined under microscopes and identified using standard guides. Data analysis were done using Shannon diversity and pollution tolerance indices. This is the water quality status ranges based on the indices. Results and discussion. Benthic macroinvertebrates assemblage. A total of 51 species of macrobenthic invertebrates belong to 3 phyla, 4 classes and 33 identified families and 2 unidentified families were recorded from Puttalam Lagoon. A total of 23 gastropod species belong to 14 families were recorded during the study period. This table shows the gastropod recorded within the Puttalam Lagoon. The highest abundance were recorded family Certipidae. Similarly, Dahanayaka and Vijayaratna found that the family Certidae common in Nigambu Lagoon and Hettige found family Certidae is the second most dominant family in the Kokilai Lagoon, Sri Lanka. Bivalve. This table shows the bivalve recorded within the Puttalam Lagoon. This graph shows the total abundance of gastropods and bivalves at each sampling location. The highest and lowest abundance of gastropods were recorded at sampling location KL16 and KL2 respectively, whereas the highest and lowest abundance of bivalves were recorded at sampling location KL3 and KL2 respectively. This is the list of polychaetes recorded within the Puttalam Lagoon. This is the list of crustaceans recorded within the Puttalam Lagoon. A total of 3 families of crustaceans were recorded during the study period. Abundance of crustacean and polychaetes. The abundance of crustacean and polychaetes were recorded at different sampling locations of Puttalam Lagoon is shown in this graph. There were no any polychaetes recorded in sampling locations, namely KL1, KL4, KL6, KL7, KL8 and KL16. Also, crustaceans were not recorded in sampling locations of KL7, KL8, KL9, and KL14. The highest abundance of crustacean and polychaetes were recorded at sampling locations KL4 and KL3 respectively. Ecological indices. This is the overall value of Shannon diversity and pollution tolerance index. Conclusion: both indices showed that health of the lagoon is in moderate condition in this rapid assessment survey. Gastropods were found as the most dominant benthic macroinvertebrates in the lagoon during the study period. This study provides baseline information of the Puttalam Lagoon's benthic macroinvertebrates. These are some of my references. Thank you.
Thank you very much, Ms. Hettige. Can I invite any questions? Okay, Ms. Hettige, would it be possible for you to comment now, for example, I noticed that the crustaceans and the polychaetes were highest in the sampling station K4, and they were not, they were totally absent in K7. What might be the probable reason? Because you say the whole, it's in moderate pollution, but pollution may be just one element that governs the distribution of these invertebrates. No?
Yeah.
There are other factors. So, are you able to shed light on any other factor that might have influenced these trends between the different samples? And how far away was one from another?
We selected actually random sampling.
Yes, but how far would one be from the other sampling station?
From KL7 to?
No. In general, I know it's random, but in general, there must be some distance, no?
Yeah.
What is the rough distance between one and another?
Maybe 1 kilometer.
They were so spread out, is it?
Yeah. This is I selected whole lagoon area.
Okay.
This is the third largest lagoon in the Sri Lanka.
Right. Okay. So in that case, other than for pollution, there may be many other different factors, isn't it?
Pollution is the aquaculture activities is happening in this lagoon.
What about the vegetation structure, the mangroves presence?
Mangroves. Yeah, it's dominant that some mangroves in Puttalam Lagoon. Also seagrass.
Yeah, so it would have been nice rather than probably you might present it elsewhere, but just for completeness, we see such a, you're saying that
sorry, madam?
No, I think someone's mic is not muted. Yeah, just to know, because you say this is moderate pollution levels. So I think there's some disturbance. Can the others mute their mics, please? Or is it your mic? Right. Just to know, because there's such a variation, it is possible that it may not be pollution at all. It may be the structure of, the sediment, the substrate, or the depth of water, or the presence of mangroves. So it would have been nice if you had touched upon that.
Yeah.
So that's for your future if you're publishing. Yeah. Okay.
Okay, thank you.
Right. Okay, thank you very much. We'll move on to the next. Yeah, so the next presentation is on the total fecal coliform and aquatic insects in Wak and Makelioya in the wet zone of Sri Lanka. And it's presented by Yasara Arachi. Over to you, Yasara.
Thank you, madam. My abstract topic is total fecal coliform and aquatic insects in Wak and Makelioya in the zone of Sri Lanka. I am Yasara Arachi, representing the Department of Zoology and Environmental Science, Faculty of Science, University of Colombo, Sri Lanka. To give you an introduction about the topic, aquatic insects play a
Can the host please
functioning. They act as
tell someone to mute the mic, please?
Sure, madam.
Is it in your area?
From the previous presenter, I guess, madam, I'll look into it.
Please, because it's disturbing. Okay, you can continue. Okay.
Aquatic insects play a major role in aquatic ecosystem functioning. They act as a controlling group in aquatic food webs and help to the nutrient retention of the ecosystems, and they help to stabilize the stream beds, and as well as they consider as a bioindicator. So why aquatic insects? They spend more than half of their life in water. They are sensitive to water chemistry, physical parameters of the stream habitats as well. Site that I use for my study. One is Walker site and the other one is Makelioya site. Walker site is a tributary of Kalni River. The population around the area is about 1,300. And one side of the stream is bordered to the Inketamuklan Forest Reserve, and the other side is bordered to the rubber plantation. And come to Makelioya site, it is tributary of Kalu River, and the population is around the area about 800, and the stream is surrounded by state forest. I use this study site as my control site. Of the research can divide into three main parts. One, the analysis of in situ and ex situ water quality parameters. The second one, the measurements of physical stream parameters. And the third one, analysis of aquatic insects and investigate on habitat diversity. 3 points in each sampling site from 10-meter distance were selected and marked for water quality checking and insect sampling. When it's come to analysis of in situ and ex situ water quality parameters, water temperature, pH, turbidity, conductivity, and total dissolved solid were measured in situ, and the preserved sample were used to measure nitrate level, ammonia level, chloride level, fecal coliform level, and total coliform level in ex situ. Under the physical stream parameters, ambient temperature, weather, shade level or depth, and flow rate, and as well as the stream characteristics such as stream width and bottom nature were recorded. The presence or absence of vegetation at the bank were hand-drawn and recorded. Aquatic insects were collected using KeepNet method and all-out search method according to the habitat characteristics, and 3 replicates from 5 important habitats were sampled. These are the results of my research. These two maps show how the land was used in two study sites. As you can see in the Wakwaya site, the land was used more for the plantation, where the land is remaining as forest at the Makelioya site. The collected aquatic insects from two study sites were represented by 5 aquatic insect orders. They are Order Odonata, Order Plecoptera, Order Ephemeroptera, Order Trichoptera, and Order Coleoptera. The below bar chart shows how the number of individuals from each aquatic insect order varies from the study site. The variation in each aquatic insect order against the collected months were studied in this research. The order Coleoptera and Ephemeroptera were observed with the same variation pattern as the order Plecoptera at Buckwheat site, and order Trichoptera was observed with the same variation pattern as order Odonata at Buckwheat site. When compare the correlation between aquatic insect orders and physical stream parameters, order Plecoptera show a negative correlation with shade level, where order Coleoptera showed negative correlation with stream depth, and as well as order Coleoptera also showed a negative correlation with the shade level at Buckwheat site. Significant association between the aquatic insects and the habitat types observed in both study sites. Order Trichoptera was observed in both leaf litter and underside of the stones in Buckwheat site and as well as in Markalier site. Other than Trichoptera, all other aquatic insect orders were found only in underside of the stones. Discussion of the research. When it's come to the water quality, it remains same when compared with the previously done study at Buckwheat site, although the water flow rate and the turbidity level have reduced. The Buckwheat study site was observed with high total and fecal coliform values exceeding the Sri Lankan standard for ambient water quality. The stream is used for washing and bathing purposes by the villagers and by the public for recreational purposes and operation of rubber plantation at one side of the stream bank. According to the previous study that have been done at Buckwheat site, the aquatic insect diversity at the site has been changed. Aquatic insects from order Hemiptera, order Ephemeroptera, and order Hymenoptera were observed previously, where order Plecoptera, order Trichoptera, order Ephemeroptera, and order Coleoptera were observed in this study, but order Hemiptera and order Hymenoptera were not observed in this current study. The acceptable concentration level of water quality parameters varies from one animal group to another. For example, some species can survive at high nitrate level, where some species cannot survive at high nitrate level. As well as some Plecoptera species were recorded to show increase in development at high total coliform and fecal coliform level. Aquatic insects are used as bioindicators of water quality from other parts of the world. Some studies from Sri Lanka also indicate that aquatic insect assemblages can be used as indicators of water quality. High diversity of species was observed underside of the stones at the river type of stream habitat within the two sites. The complex habitat structure of stones, which can trap litter and woody debris, provide food sources and shelter, is the main reason for that. The presence of freshwater fish at Buckwheat site encouraged the habitat diversity underside of the stone as well. The conclusions of the study are: aquatic insects were significantly associated with the habitat type they occupy. Total coliform level was unacceptable for ambient water quality standards of Sri Lanka in Buckwheat site. The presence of fish, including several endemic species, was found from stream at Buckwheat site, despite the fact that there is significant fecal coliform pollution. This needs further investigation from other parts of Sri Lanka, where endemic fish are found in land uses with high level of human activities. The need of regulate the human activities as well as taking measures for protection of freshwater streams is highlighted in this study.
Thank you very much, Yasara.
Thank you very much.
Right. So once again, the floor is open for any questions. I can see the architect, Mrs. Sherina Amendra joined in. Madam, do you have any questions for her?
Thank you, Dr. Mayuri. I won't have any questions from this. Thank you very much.
All right. Can I invite any other participants? I'm forced to ask questions as the chair, but if anyone else can help me out. All right, Yasara. I saw you had mentioned, at least in your abstract, that the fecal coliform count was high in site 1, is it?
Yes, madam.
Yeah.
Buckwheat site, yes.
Right, and that in the second one, the diversity might have been high due to in number 2, site 2.
Yes, yes,
Markelli.
Yes, madam. Yes, Markelli site, yes.
Yeah, but if you look at the Plecoptera and the Ephemeroptera, their diversity or species richness rather is much lesser than in the other side, whereas this EPT is known as the index for that can be used to indicate pollution. Would you have any explanation for that? Why Plecoptera and Ephemeroptera were low in site 2?
Madam, it may be due to the stream run like the water running fast in the Markelli site than the Buckwheat site, and the rivers are more prominent in the Markelli site. I think that the flow rate can be the big reason for these changes.
And you had also mentioned that it is associated with the habitat type. What exactly do you mean?
Madam, like I studied whole different kind of micro habitat like sand, the riparian vegetation, and underside of the stones, and in the debris as well. But when I compare all of them, most of these aquatic insects are prominent at the underside of the stones. Like in sand, I couldn't find any kind of aquatic insect samples. In other debris as well. But the underside of the stones, they are like more prominent underside of the stones.
Right. So is it possible that the second site did not have any of these preferred habitats? Microhabitats? In if stones are so important, maybe if there was a diversity in the distribution and abundance of these little pebbles or stones between the two, and also possibly other parameters like water depth might have also contributed to the variation, right? Yasara?
Madam, I didn't hear you.
Yeah, in addition to this fecal coliform, as you say, the abundance and distribution of pebbles or stones and the depth, water depth, might have also affected it, right?
Yes, madam. Yes, that can affect.
So the conclusion drawn possibly that these two might be because of fecal coliform may not be that credible.
Okay.
Isn't it? I don't know. I'm asking you. You're the one who did the study.
No, madam. I didn't had much time to sample. If I did more samples,
no, that's okay. That's okay. This is a frequent thing that everybody says no time, but that's fine. From what you gathered, what I'm saying is you would have studied some water parameters and some habitat parameters.
Yes,
that you consider this fecal coliform as just one, and also incorporate the other factors into the model, so that would give you as to what is the most overriding, which factor had made the greatest contribution to changes, right? So that is for the future. Yeah, if you're doing a paper or something.
Yes, may I make an observation?
Yes, please, of course.
I think the papers have to start with certain assumptions that focus is being taken only on certain parameters, because aspects like snags, stream bank character also has a contribution. So perhaps the papers should focus on a particular subject, having made certain assumptions or exclusions at the beginning of the paper.
Yes, I agree with you, madam, but there are certain fundamental parameters that can be measured.
Measured, yes.
Yeah. Now, as you say, the morphology of the stream bank and all, maybe it is assumed that both would be similar, but there are like water depth and flow are two key characters that can easily be measured. So anyway, that's food for thought for you, Yasara.
Yes.
Thank you.
All right. Okay. Thank you, madam.
Thank you, madam.
So we'll move on to the next presentation, which is a systematic review on effects of global climate change on birds, and that will be presented by HMS Fernando.
Hello everyone,
Alicia
I'm Alicia
Fernando.
Fernando and I will be presenting. Hello
Go ahead,
everyone,
yes.
I'm Alicia Fernando and I will be presenting the systematic review on effects of global climate change on birds. The corresponding author is Mr. Medisha Gunawardena. These are the content that I will be covering in my presentation. Climate change, on a whole, is defined as the change in the state of climate that can be identified using statistical tests and that persists for an extended period of time. The consequences of this global problem is affecting plants and animals immensely. Out of all these animals and plants, birds are highly mobile creatures that are very easy to observe. Therefore, the consequences of climate change on birds is one of the best documented changes reported in the animal world. Although birds are highly studied organisms, we do face challenges in acquiring knowledge. If we are making plans to conserve a specific bird population in a certain location, we have to make predictions and projections of their behavior to make conservation measures successful. It is mandatory to assess knowledge and predict the effects of climate change to avian communities. Our major objective was to review the studies conducted in the two most recent decades regarding the topic. We highlighted the number, information, and patterns of research and also highlighted the importance of projections and predictions. This study makes it evident that research of this nature is important. It identifies gaps and limitations and also provides recommendations for future research. The methodology used in the study is a standard systematic review methodology that can be used for environmental studies related reviews. Here, as you can see, population, intervention, comparison, and context are subdivisions of protocol. Search is subdivided into strategy and delivery. As I explained earlier, in order to identify the scope of the review, also known as the protocol, we have used the PICOC framework. It indicates the population, intervention, comparison, outcome, and the context of the review. Moving on to the search strategy, two databases were used, PubMed and Google Scholar. Here, as you can see, the article availability in both databases. Once extracted, here's the number of articles that were taken for the review. In total, 170 articles were extracted. When we extract data from such a huge amount of literature, a proper criteria is a must. These were the 10 criteria used in order to extract data and the categories considered. Let us have a look at the results. There was an increase in the number of research in both databases that were used. The highest number of publications were available in 2019. The most published type of article were journal articles. The highest occurring journal was PLOS 1, and the selected articles ranged between 1830 to 2100. 28 studies were done in a global scale, while other studies scaled around 8 regions, 5 continents, and 40 countries. The articles were categorized according to the type of climate change consequences on birds. These were the focus areas that were identified accordingly. The articles that used primary and secondary data sources are more or less equal. Quantification, mapping, and modeling were the most used assessment types. The most studied species were the pied flycatcher and the blue tit, and the majority of the publications were based on the purpose of expanding knowledge. Let me take you through the most important criterion of the review, the focus area. The graph on the left shows that most studied focus area is ecology, while the least is evolution. The chart on the right shows the subdivisions of each of these focus areas. Behavior has 4 subdivisions. Migratory birds may completely stop being migratory due to climate changes and therefore change its status to being resident birds. Then they become a competition to the birds in the area. Global warming may have a positive impact on short-distance migrants. However, long-distance migrants are negatively affected as they rely on the photoperiod as a cue to start migration. Birds are usually adapted to migrating along the wind direction to avoid unfavorable conditions. When climate change affects wind direction and speed, it affects these migratory populations. There is a specific timing for the arrival and departure from breeding grounds. These timings may also collapse due to climate change. For example, the American robins that initiate spring migration to higher elevations earlier respond to environmental cues in the overwintering grounds. They arrive 14 days earlier to the breeding grounds than 20 years ago. Egg-laying patterns have 2 subdivisions. 60% of the studies say that there is a long-term advancement in the laying date. When birds become non-parallel with the natural availability of food, there will be a negative impact on food supply and ultimately on egg-laying patterns. Temperature is the major factor deciding the viability of eggs. Warmer springs may result in smaller eggs and also smaller clutches of birds. There are 3 subdivisions under feeding pattern. Although the conditions are favorable, some birds prefer to wait until their favorite food springs in. This can cause a delay in the arrival and departure times. Also, phytoplanktons, which is the basis of all food chains, are being affected due to global warming that causes stratification of ocean surfaces. This affects the food chain, the ecosystem structure, and ultimately the feeding patterns of birds. There are 3 subdivisions under nesting behavior. Heavy rainfall patterns can reduce the foraging time and floods can destroy the bird nests. Sea level rise can cause either a habitat change or a habitat loss. Wildfires can directly affect bird populations by completely destroying the habitats. Moving on to physiology, birds that have low basal metabolic rates experience limited temperature variations, but global climate change has a huge temperature variation that has negative effects. Normally, birds may change their body size or other morphological traits as a response to climate change, which relates to thermoregulatory issues. If temperature continues to increase at this rate, it is important to monitor them because the species ranges are already contracting in certain bird communities. Climate change can also lead to directional selection, which is a type of natural selection in which the phenotype of the species tends towards one extreme rather than being in the mean phenotype or the opposite extreme phenotype. Migratory mammals can be resilient to climate change if they can shift their ranges. They can use their phenology to track the suitable climate change only up to a certain extent. Onset of breeding is affected by factors such as temperature, precipitation, and food supply. In a certain study, 45 out of 57 species advanced their onset of breeding. Also, in evolution, birds such as blackcaps that breed in the southwestern Germany arrive early, not only because of a high breeding success, but also as it drives associative mating among the males with comparable timing. This results in a unidirectional evolution, which is a negative aspect. In conclusion, although research based on climate change consequences are rising, more research should be done in the areas of less focus, especially in Sri Lanka. These are the references that I have used in the presentation, and my heartiest gratitude to all those who helped me in making this review successful. Thank you.
Thank you very much. Once again, I have to ask whether anybody wishes to ask a question or a clarification. All right, Ms. Fernando, can I ask you why now climate change studies, you need data from a fair length of time, isn't it, to arrive at a conclusion that the changes are indeed because of changes in climate? In what now, were many of these experimental or were they were they kind of in the field-based studies?
Good afternoon, everyone. Thank you so much. In the review, there are both studies which have been using long-term data, and also there are studies which have been conducted by using other studies, which means secondary data as well. So most of the studies actually were conducted by using data that were collected for a lengthy period of time.
All right. Can I also ask you, that is one of the difficulties in attributing some observed change to climate change, because lot of other things also have changed. Now, for example, the habitats have undergone severe or considerable changes across the years. So have any of these studies kind of stripped away those factors before attributing something to climate change? Say, for example, the non-arrival of migrants might be due to some changes in, or maybe the non-arrival of turtles, maybe because of the changes in thermal currents which have resulted from climate change, right? But it might also be because of the lack of the particular beaches or the destruction of those particular beaches. So likewise in birds, also there can be other overriding factors. So what's your opinion on that?
Yes, so I actually came across few studies that took into consideration those factors. For example, the breeding, onset of breeding, there was actually a study which showed that there were destruction to the environment which could have caused a later onset of breeding. But that was only for few studies. Most of the studies did not take many factors into consideration, which means they only took the climate change data and then directly attributed it into the behavioral changes. So that is something we could really improve, and yeah.
Okay, it is difficult to do, I understand.
Yes,
but
but it could improve the quality
of
exactly.
Yeah, it can. It can, yeah. And another small thing I was going to ask is that you had in your abstract, you had said that studies on the genetic changes and evolutionary changes and physiological changes have been studied, on a limited scale, but that is most likely because those require invasive procedures, whereas observations on behavior or ecology can be done without any direct capturing of birds, etc., isn't it?
Yes, but the genetic studies could actually lead to more a deep understanding of the behavioral changes. So,
true, exactly. Yeah, but maybe that might be one of the reasons why these are not done. So we conclude our session. Can I just find out if Miss Tadani is logged in?
Madam, she has mentioned that she wouldn't be able to come today.
Okay, all right.
Madam, we can proceed to the evaluation.
Yes, that's so. Hang on for a minute. I will.
Yeah, sure, madam. Sure, madam. Take your time.
Yeah, the best presenter. So please be logged on.
Sure, madam. Take your time. Thank you very much.
Okay. So it is my pleasure to announce that the winner or the winner of the Best Presenters Award today goes to HMS Fernando, who made the presentation last of all on the systematic review on effects of global climate change on birds. So congratulations to you, Ms. Fernando.
Thank you so much, madam.
All right. So I take your leave. Is there any other task to be done? Can I ask the coordinator of the session? Yes. I have concluded the session. Is it possible for you to formally wind up, or shall I do?
Yeah, sure, madam. I'll do it.
Okay.
Madam, could you please send the evaluation
Right, I will.
Sure. Thank you very much, madam. So we have come to the end of this session. And thank you very much, Professor Mayur Vijay Singha, for chairing this session and for the immense support you extended towards us. Also, I take this opportunity to thank our keynote speaker, Professor Cyril Vijay Sundara, for joining hands with us today. And we are so grateful to have you with us. Finally, I'd like to thank all the presenters for joining hands with us, and we wish you all the very best for all your future research and findings. So have a good day. Thank you very much for joining with us.